Bismillahirrahmanirrahim. Welcome again, guys. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about the B cell activation and the B cell differentiation in detail. In the previous video lecture we talked about the B cell development and B cell maturation, as well as we talked about the B cell biology overview, and we talked about the B cell receptor in detail. If you missed these video lectures, so go back to my channel and watch this video lecture. There will be a playlist present on the B cell, so then go back and watch this. Once you get idea about that, so then this B cell activation and differentiation will be very easy for you guys. So without wasting the time, let's start the video. Okay, friend. So we are going to talk about the B cell differentiation and activation. So remember, guys, first of all, let me tell you a little bit idea about the B cell activation and differentiation. Then we will explain it with the help of diagram. You know, B cell will be activated with the help of antigen fragment or the fragments of the microbes when it bind on the B cell surface receptor, which we call a B cell receptor, or we also call that antibody of the B cell. When it bind on the B cell receptor, so then what happen? This antigen actually provided signal to the B cell. The fragments which are released out, it is actually released out from the neutrophil. The fragment which is provided to the B cell, it is actually released out from the neutrophil. Neutrophil, first of all, they will engulf and then they will release the fragments of the microbes. After that, once the fragments are received by the B cell, any one of the B cell, because we have different B cells having same type of the antibody on their surface. When they receive it, so then signal is provided inside the B cell. Due to this way, the B cell will be enlarged, and they will enlarged and converted into plasma cell. Due to which we can say after that the B cell will be activated in form, and this activated form of the B cell converted into plasma cell. After that they will secrete antibody. Those antibody due to which the antigen are captured. Okay, so now let me explain with the help of diagram. So let me write B cell activation. Remember, guys, B cell will be activated with the help of two way. Okay, it will be activated either by T cell activation, or it will be activated by without T cell activation. So let me write T cell develop T cell activation, T cell dependent B cell activation, or it will be activated by T independent B cell activation. What does it mean? It means that when the B cells are activated, it requires the T cell activation. Sometimes the B cell activation doesn't require the T cell activation, and without the T cell activation the B cell will be activated. Okay, now how it requires the T cell activation? So now the T cell dependent B cell activation, in this case we mainly require three types of the cells. Let me write. We require B cell. First of all, we required macrophages, and we also required the T cell. In T cell we required helper T cell, and in helper T cell we required helper T cell 2, and moreover we required B cell. So these things we required for the B cell activation. Now what happened for the B cell activation? First of all, the macrophages will engulf the microbes. Okay, for the B cell activation, first of all the T cell should be activated. Once the T cell is activated, then the B cell will be activated. Okay, so what happened? Let me explain. First of all, we have the microbes. As you can see, this microbes is actually engulfed by macrophages. So let's suppose this is the macrophages. Once they engulf it, so then these microbes are break down into small fragment, as you can see, and after that one portion or some portion of the microbes are then presented to the receptor of this macrophages. So let's suppose this is the portion of the microbes. Once this microbes is presented to the receptor, this receptor is called as MHC2 receptor. Now this MHC2 receptor actually processing, or they actually presenting or express the antigen fragment or the microbes fragment of to their receptor. Okay, once they representing it, moreover they also produce some kinds of cytokines which we call that interleukin 4. This interleukin 4 just attract the naive T cell. So after that we get the naive T cell. So after that what we get the naive T cell. So let's suppose this is what, this is the naive. Okay, after that we get the naive T cell. This naive T cell will also recognize this one receptor. They also recognize the macrophages via the antigen. Okay, and moreover this naive T cell have also the CD4 molecule. This CD4 molecule actually properly places this T cell receptor to this MHC2 molecule to recognize the antigen completely or fully, we can say like that. This is the CD4. After that, this B cell have also another co-receptor molecule which we call that B7. So this is B7. This B7 co-receptor molecule of the macrophages is recognized by the CD28 of the T cell. Okay, so this is CD28, this is B7. Both are recognizing with each other. After that, the crosstalk between macrophages and the T cell occur, and due to this way, this interleukin 4 are fall down on the helper T cell, and this T cell is converted to helper T cell, which we call that helper T cell 2. After that we can say that helper T cell 2 are formed and it become activated. Now this activated form of the helper T cell 2 will bind to the B cell after that. Okay, so this is how the T cells will be activated. I already explained that T cell activation in detail. This is just overview. This is just a short concept about the T cell activation. If you guys want to watch about the T cell activation in detail, so go back to my channel and watch it. Now once this T cell are activated, so then it shifted and bind to the B cell. On the other hand, what actually in the previous video, if you guys remember, we talked that we have also the B cell. Let's suppose this is the B cell. This B cell having the receptor, so these B cells have also the receptor. Okay, so remember guys, what happened in this case? In this case, if you guys remember in the previous video, we just talked that the B cell are just selected and mature with the help of antigen portion. Okay, so this is also the antigen. What happened? Sometime if the microbes are bind to the B cell receptor, So this is actually the B cell receptor, okay? This black one, and this red one are actually the antibody. So not here, if these microbes are bind to this B cell receptor, let's suppose. So then what happens? They will engulf it, break down it, and present it to their receptor. So what we get idea about this? We get idea that this B cell is also what? This is also the antigen presenting cell, just like the macrophages. Remember guys, okay? Macrophage is also antigen presenting cell, B cell is also antigen presenting cell. This sometime what happen? This B cell actually received the fragment of the microbes from the neutrophil. On the other hand, you know, if in the previous video, if you guys remember, we also draw the neutrophil. When the neutrophil engulf the microbes, so let's suppose this is the microbes. When they engulf it, and then they also break down these microbes into small fragments and then release out from their body, as you can see. And then one of the fragments of the microbes bind to this. After that, this fragment is bind to this receptor of the B cell, as you can see. Once it binds to the receptor of the B cell, after that this B cell will be activated. Sometimes this phenomena occurs. Sometimes what happen? This B cell will engulf the microbes. They also engulf the microbes by themselves, and they doesn't require the neutrophil. When they engulf the microbes and presented it to their receptor, after that what happened? The T helper cell 2, which is in activated form with the help of macrophages, they will contact with this B cell. This is B cell. And what happened? They recognized this one receptor molecule, which is MHC2 also, MHC2. So let's suppose this is the T cell. And this is the T helper cell 2. Cell 2, okay? Now they recognized this one receptor, MHC2 receptor, by their receptor, which we call that T cell receptor. Okay? When they recognize it, so they recognize this MHC2 receptor with the help of T cell receptor, and moreover, they also required another molecule which we call that CD4. Okay, so this CD4 actually properly places this T cell receptor to this MHC2. Moreover, we have some other receptor, co-receptor molecule recognition are also done. Means this B cell also, you know, producing another co-receptor molecule which we call that CD40 lignin, which is recognized by this T cell receptor, which is recognized by CD40 of the T cell. So this is CD40 of the T cell. Once all this recognition is done, so then what happened? The cross-talking between the B cell and the T cell is started. When the microbes are bind, so it doesn't mean that the B cell is activated. Still the B cell is not activated. It required the T cell, activation of the T cell. When the T cell is activated and binded, so still it is not activated. After that what happened? This T cell will secrete some interleukin, as you can see. They will secrete what? They will secrete interleukin 2. Before that, you know, this helper T cell which bind to this macrophages, I forget a very important things here. When they bind to this macrophages, so then this helper T cell will also secrete interleukin 2. And this interleukin 2 will bind to the receptor of interleukin 2 receptor. You know, this, once the recognition is done, so then there is chromosomal changes or transcription translation will occur, due to which interleukin 2 receptor is formed, and interleukin 2 cytokine is produced and again bind to this one cell. When it's bind to this one cell, so this cell is then activated, and this cell is then converted into, or we can say we get a lot of T cell. So we get a lot of copies of the T cell, helper T cell 2. Among them, some we have effector T cell, and among them some we have memory T cell. Okay, and after that some of the T cell will be shifted and bind to this B cell, and all this process will be going on, which I explained earlier. So when the recognition is done, they will secrete interleukin 2, which is received by this B cell via the B cell receptor, B cell interleukin 2 receptor of the B cell. This recognition actually brings some chromosomal changes inside the B cell, due to which interleukin 2 receptor is formed, and after that interleukin 2 cytokines are released from the T cell, and it binds to the interleukin 2 receptor of the B cell. Due to this way, not interleukin 2, sorry, interleukin 4. This interleukin 4, once bind on it, so then what happens? They will cause the B cell, a little bit they will cause the B cell growth. So after that what we get B cell growth. So this interleukin 4 is actually acting as a B cell growth factor, B cell growth factor. It means that this B cell will be a little bit enlarged, and the chromosome means the cytosolic portion is enlarged. The nucleus portion get decreased in size, okay, and due to which we get a plasma cell. But this is not enough. After that, there is another interleukin is also secreted from the T cell, which we call that interleukin 5. This interleukin 5 will also bind with another receptor on the B cell, which we call that interleukin 5 receptor. It bind on it. After that what happen? This B cell will be expanded, or this interleukin 5 is actually acting as a B cell expansion factor. It means that this B cell will be expanded, and moreover this B cell will be converted into different, or we get a lot of this same B cell. So we get a lot of the same B cell. What does it mean? It means that this is that type of the B cell which have the IgM or IgA or IgG, whatever you can say, which have that type of the antibody, and that type of the antibody is actually that type of antibody actually capture the antigen. Let's suppose the antigen is captured by the IgE antibody, so we get a lot of B cell having IgE antibody. And the neutrophil, this interleukin 4, this interleukin 4 is also bind to the rest of the B cell which are matured. Now once these mature forms we get, so then the interleukin 4 will also fall down on each B cell. From each B cells, it will also cause some changes inside the B cell, which we call that cytosolic enlargement, and nucleus portion decreases. Due to this way, we get the plasma cell. Among each B cell, what we have also the memory B cell also. Memory B cell. So we have also memory B cell among them, and we have also the same type of the B cell. And all this B cell will be converted into plasma cells. So then let's suppose this is what? This is the plasma cell. So what we talked that the nucleus get decreases. So this is, let's suppose, the nucleus, and moreover the cytosolic portion enlarged, and we get a lot of antibody in this cell, and this cell is called as a plasma cell, because all of you better know plasma cell is nothing but a kind of antibody producing cell. And remember guys, these are those antibody which are earlier captured by the antigen. Let's suppose these are the antibody IgE which are captured by the antigen. So that type of the antibody will be making inside this plasma cell, and then they will produce each type of antibody against this antigen which is present inside the body and kill it by itself. If you guys remember, I already talked that these antibody have sometimes the ability to kill the microbes alone, and sometimes these antibody will just coated the microbes and due to this coated of the microbes, what happen? Opsonization process will occur. It means that macrophages and other antigen presenting cell or neutrophils will come, and these microbes will be easily engulfed. Okay, sometimes microbes get filled to engulf, sometimes this antigen presenting cell or neutrophil are filled to engulf the microbes. So then antibody will then bind on the microbes, due to which the neutrophil and antigen presenting cell will easily engulf it. This is how antibody are very important. Sometimes these antibody are specially responsible to perform the, what we can say, complement system or complement process. I already explained the complement process. Okay, sometimes these antibody are specially responsible for the complement process as well. So this is all about the B cell activation and differentiation with the help of T cell dependent B cell activation. So this is actually the B cell is activated, but with the help of T cell activation. As you can see, T cell is activated, after that it activates the B cell. Next we have the T cell activation, the B cell activation without the T cell, means T cell independent B cell activation. It means that the B cell doesn't require the T cell activation. B cell doesn't require the T cell for their activation. What happened in this case? In this case, if we have the microbes, all those types of the microbes present inside our body, which have the Lipopolysaccharide, and we have glycolipids. If we have tumor promoting antigen, nucleic acid. If you have these types of the molecules on the microbe's surface, so then this B cell doesn't require what? This B cell doesn't require the T cell. What happened? Let me a little bit explain this one phenomena. They have also, at this stage we have also the microbes, as you can see. Now this, on the surface of these microbes, there will be one molecules. Either they have lipopolysaccharide, glycolipids, or the rest of the things. They have just one. Or sometimes they have also all these things as well. So let's suppose these are the antigen, as you can see. And these antigen are this type, let's suppose. Okay. So then what happened? The B cell receptor will come contact and recognize this one antigen, and sometime they will also, this B cell have also the toll-like receptor. If you guys remember, we already explained the toll-like receptor. These B cells have also the toll-like receptor, and they also recognize the rest of the antigen. So remember, if the B cell recognize the antigen, which is the lipopolysaccharide glycolipids, and the toll-like receptor is also recognizing the same molecules present on the B cell, present on the microbes. So then what happen? This B cell will get activated. After that, this B cell will get activated, and once it activated, so again it converted into plasma cell. And again the rest of the process will occur, which I already explained. Means it again converted into plasma cell, and the plasma cell will secrete the rest of the antibody which recognize that type of the antigen, which recognize that type of the microbes. So then they will secrete these types of the antibody. Okay, but remember guys, in this case no memory B cell formed. In this case, no memory B cell is formed. In the T cell dependent B cell activation, memory cells are formed. But in this case, memory cells are not formed. Memory B cell are not formed. Okay. So what we learn? We learn that this B cell doesn't require the T cell activation. As you can see, there is no T cell due to which the B cell is activated. B cell is just activated to recognize these types of the antigen molecule which is present on the microbe surface. After that, the B cell will be activated, and it will be converted to plasma cell. Plasma cell will secrete antibody which are against to this type of microbes. So this is all about the B cell activation and their differentiation. I hope you guys get idea about that. If you like this video, so make sure to hit the like button, share this video, and definitely subscribe the channel to get more interesting videos like that. Thank you so much for watching.